next speaker is Rozman, who will tell us about extensile-contractile phase separation in mathematics.
Okay, thank you very much for the introduction and for the wonderful conference. So my talk will be a bit of a continuation of what Julia presented at the start, as I've been essentially discussing how I get phase separation between active nematics of different activities, but this time zoomed in more at the level of individual cells. Now, as we already had enough introductions to this, so I'll just go very quickly over what the active nematic story is. The idea is we have some sort of nematic system, and the individual components generate the usual Sima-Ramaswamy active stress. So zeta is the activity, Q is the usual tensor to describe nematic order. If zeta is negative, then it's contractile, and the active flows are inwards along the long axis, outwards along the short axis. And if zeta is positive, it's the opposite, and they're extensile. Now, one of the systems that seems to be exhibiting nematic order, and for which nematic activity seems to be important, is actually epithelia. This is a type of tissue that's very important in development because it's a key component of early embryos. It also covers the majority of organs, and it consists of prismatic cells sticking together. So what we wanted to say is, what we're interested in is, okay, if you're modeling epithelia and you want to model them at cell level resolution, one of the standard approaches for this is what's called the vertex model. The idea is, well, if you look at these cells in a monolayer, they more or less look like polygons, right? So if you want a model that's computationally efficient enough that you can simulate thousands, tens of thousands of cells, but on the other hand, it has enough geometric information about each of them, then the easiest way is, okay, let's just say that each cell is a polygon. It's defined by the position of its vertices. And then the usual assumption is that these vertices move with a velocity proportional to the force acting on them. The argument being, this is a low Reynolds number system. There's friction, so this gives the equation. Then that consists of, well, the system is trying to minimize some passive energy, and it's experiencing whatever active forces you end up putting on top of this. And the passive energy tends to just be, well, each cell has some area it wants to have, which tends to get reabsorbed into being the unit for making it non-dimensional, and each cell has some perimeter it wants to have, and a target perimeter. And deviations are penalized by harmonic terms. And famously in these models, as P0 increases past a critical value of around 3.81, they go from having a solid-like response to having a fluid-like response in terms of shear and in terms of barriers for topological changes. Now lastly, what we need to add to the model is topological changes. So for example, if a junction between two cells becomes very short, the network gets restitched so that the two cells that previously weren't in contact now are, which in practice ends up looking like this. Okay, so what we want to do now, this is sort of one of the standard models for epithelia. And if epithelia are active nematics, well, how do we add nematic activity on top of this model? What do we need to get to add to get the correct behavior, and what's missing? Now, there were actually two papers that came out on how this can be done, one by the groups of Jean-Francois Pré and Matthias Merkel, and one by us. And the one I'll be focusing on here is actually the one from the Arx-Berthet groups, because it's closer to phase field models, and things I'll be talking about, we first tried doing phase field models. So their idea is, well, if you have some unspecified stress tensor sigma, and you want to just figure out what are the forces on one of the cells, so one of the polygons, you can go through this procedure. Well, it's easier to figure out what the force is going to be on a junction. It's just going to be the dot product of the normal of the junction and the stress tensor. And then because it's a vertex model, so what's actually moving around is the vertices, you have to in some way map the force on the junction to being a force on the vertex. The idea is, well, let's just say that each vertex holds half the force acting on all of its junctions, which gives you this, and with some math you end up with this equation. Now, the model they proposed was that if you want to do an active nematic with this, let's just say for each cell, you can define a Q tensor, which now doesn't describe sort of the local averaged out nematic order, but the direction of elongation of that specific cell. So this is just the perimeter of the cell, the length of the junction, the unit vector along the junction, and this just makes it traceless. And what they found was that as you increase the activity, so as this model is extensile, for sufficiently high extensile activity, it's going to become something like an active fluid. It's essentially exhibiting active turbulence, and if you look at the defect dynamics, it looks more or less correct in terms of how comet defects move. Now, the model does only work for extensile activities and for contractile activities, it just remains solid because the difference is here the cells are effectively squishy, which means that if they are generating contractile forces, they're going to round out and then they're going to stop generating additional forces. Okay, yes?
Does the only place that the Q tends to enter in the active stress, does
Yes.
it not enter anywhere else?
Nowhere else. It's just the standard vertex model, so area and perimeter elasticity on the cells, and then you put active stress, which is the usual equation. It's per cell, and then by the scheme on the previous slide, you translate it into vertex forces. And it looks approximately like an active nematic for high enough extensile activities. So what we're talking about is sort of adventures in trying to map this to continuum results and seeing sort of what else needs to be added to get the dynamics you would expect from the continuum models. The talk will basically have two somewhat connected parts. The first one will be how do you generate flows with it, and the second one will be going back to active nematic phase separation. So how this started was we wanted to see, okay, can we map the model onto the continuum equations? And one of the lovely things about active nematics is if you confine them in a channel, they're going to have a well-defined sequence of flow profiles as you increase the activity. So they first generate unidirectional flows, then oscillating flows, then a dancing state, and then active turbulence. So the idea was, okay, if we can map this behavior to behavior in an active nematic vertex model, that would be a way to relate it to. So this was our model. We have a channel, there's periodic boundary conditions left and right, mostly boundary conditions top and bottom. The red lines just show the long axis of the cell, and a handful of the cells are colored so that you can track movement. And we found that nothing we could do with this model would actually make it flow down the channel. Essentially, it always just exhibits active turbulence. It never generates any other type of motion. And this was completely irrespective of the activities. As long as it's an active fluid, it was always turbulent. So we were a bit confused by this because, well, this is a pretty good way to implement active nematics at the cell level, and the active nematics really should be able to flow down the channel. So we started thinking, well, when you're doing vertex models, pretty much universally, the equation of motion you assume is this, that the force is proportional to the velocity. This is a dry model. It's so substrate friction is what's dominating the dynamics. On the other hand, the continuum models are Navier-Stokes. In low Reynolds numbers, what's balancing out your active and passive forces is viscous dissipation as opposed to frictional dissipation. So can we do something like a wet vertex model and see if that fixes our problem? Now, this equation was, to the best of my knowledge, written down for the vertex model by our collaborators in Raskos-Knepnik's group in Dundee. And the idea is, let's add an additional term. So we have the usual, the velocity of the vertex I, and its force, the force on it, and then we have this thing which is essentially drag between the vertex I and the velocity of all of its neighbors. So what you end up with is a set of coupled equations for the motion of each of the vertex, and you need to solve that on every step. And then if you go into the limit where eta is much smaller than psi, you end up with something that's essentially a wet model. You still need to leave a little bit of substrate friction in just for numerical stability, but essentially get very close to a wet model. So then we went, okay, let's do the exact same channel, the exact same activities, but now we're in the limit where internal dissipation is the dominant one. And it completely changes the dynamics of the system. You very neatly get unidirectional flows going down the channel. So it does seem to be working that just accounting for viscosity, which is to some extent obvious, friction screens flows, but it's a thing that sort of needs to be added to the models if you want to do active nematics with them. Okay, then the next thing is just quantifying this.
Best what value of parameters you had in that simulation of the model?
For the channel, it's
For the area and the perimeter to square root of area ratio. Target perimeter.
here it's 3.85, but it works 3.85, but it works for 1, it works for 4. So
I've never seen such elongated.
yeah, the extensile activity stretches the cells a lot. Like if you measure the Q, it's going to be 4. Like the measured perimeter by square root of area is 4.7 to that effect, because extensile activity, essentially there's the forces of the extensile activity elongate the cells a lot. So even if you go way in the solid regime, like P naught equals 1, at the activity at which it fluidizes, it becomes very elongated. So you essentially can't have rounded out cells in this model, which are at the same time being an active fluid.
Sorry, what is the hydrodynamic length scale here compared to the channel width? So
We'll,
velocity divided by
yeah, we'll get to that. Quite high, actually. But, yeah.
so the vortex size is less.
Yeah, it seems that it goes to trying to do the dancing state as you increase friction. So it's doing roughly what you would expect. Like the length scale seems to be quite a bit higher than the width of the channel here, which we're a bit confused by, that we're still working out. But okay, yeah, the first thing is just quantifying what happens as we go in the channel from the wet to dry limit. One thing is, in the dry model, when you look at transport, it's diffusive, it can be super diffusive, but it's never really ballistic. And in the wet model, you just get nice ballistic transport down the channel. Then this is just the velocity versus the activity. You have a threshold. This isn't the usual threshold activity you would get in the continuum. It comes from the fact that this model has what's called a rhombile state at very low activity, so it becomes essentially crystalline. And last thing is, as you increase friction, the transport down the channel goes away, and you go back to having active turbulence. And this does seem to do roughly what you would expect, which is that as you increase friction, you get more and more vortices as opposed to just transport down the channel. And also you can just plot how does the average velocity in the channel look like. For the dry model, it's essentially chaos as you would expect. And for the wet model, you get, what you would expect. The usual velocity profile, which looks roughly like Puisel flow going down the channel. And superimposed are the average shower directors. Now, when this happens is, well, as you go into the wet model, you can start getting long-range correlations. Because if you look at the dry model and you calculate velocity-velocity correlations or director-director correlations, they end up being essentially 1 to 2 cells long. But as you add viscosity, you start and you reduce the substrate friction down by orders of magnitude. Your length scale starts to become very, very long, and eventually you have to simulate something of 40,000 cells to even be able to measure it. But you end up with length scales which are of order 40 cells. So twice the width of the channel, I think, is roughly where we are. Okay, yes?
So in your model then, would it be fair to say you can map your active stress to something which is not spatially correlated when you put it in? Is this coming out, or is there any internal expected correlations with the Nematic?
So you mean like the nematic order, I think, comes from the flows, because if you have, there's nothing in the passive vertex model, as best I can tell, which would be aligning your directors. You can have zero energy in the passive model if you're in the fluid phase and have completely disordered director profile. So what's giving you alignment is the flows, it's not the passive energy. There's no KQ term in your energy, essentially. Okay. So this was sort of the first part of this, which was adventures in trying to get sustained flows in the vertex model. And then the second part is going to be trying to use this to do phase separations. Now, Julia already mentioned that if you have a two-fluid mixture of active nematics, where one is active and the other is passive, or they just have two different activities, that's going to drive phase separation. So we wanted to see, okay, can we do this at the cell level in the vertex model? So this is our setup. We have a 50-50 mixture. Orange cells are contractile, the blue cells are extensile, the white lines show the long axis of the system. And right now we are at P naught is 3.95. And what happens is the system is going to perfectly phase separate over time. So you end up with one region, or connected region of all the contractile cells, and one connected region of all the extensile cells. To quantify this, we can measure the sorting parameter, so the number of like neighbors of a cell by the number of like plus unlike neighbors of the cell, and here it's plotted over time. So as you increase, so it goes quite slowly, but it does eventually reach 0.9, so the system does phase separate fully. Now, if we look at like how the final sorting index depends on the extensile and the contractile activity, we find that here we plot the contractile activity versus the sorting index for different extensile activities. And it turns out that the dependence on contractile activity is monotonic. So the more contractile it is, the better it phase separates. On the other hand, the dependence of extensile activity is non-monotonic. So you need some extensile activity, or it's just not going to phase separate. And this is essentially because the extensile cells are motile and the contractile cells are not. So if there's nothing moving them around, it just has no way of sorting. But on the other hand, as you then increase the extensile activity, it starts to, there's sort of an optimal value past which increasing it further actually starts to adversely affect your phase separation. So now, this story actually also works if you go into the solid phase of the vertex model. So if you have sort of like P naught equals 1, so at this point the passive model is completely solid. It's still going to phase separate slightly less well, and it still has a monotonic dependence on contractile activity, and it still has a clear optimal value in extensile activity. You now also get a threshold. I mentioned this before, but in this model, if you're in the solid regime, there's a threshold value of activity for it to start flowing, and that value more or less matches the value where you will start seeing phase separation. So nothing very surprising here. Now, there seem to be two mechanisms which are driving this process. The first one is that, well, if you're looking at a pure extensile system, it's motile. The system is flowing, it's doing active turbulence, and if you just measure MSD, it's going up in time. On the other hand, a pure contractile system isn't motile. The cells essentially fidget a little bit around their center, but they're caged by their neighbors, they're not rearranging, there's no motion. And then if you mix them, it turns out the extensile cells are still going to be much more motile than contractile ones. So, and differences in velocity is one known mechanism for getting phase separation. Now, this is not the only mechanism for why this is happening, and we're not completely sure what's driving the other one, but it's essentially that contractile cells stick together. If you start sort of having a mixture which has extensile activity and contractile activity the same and fairly low, and you have a small droplet of contractile cells in an extensile bulk, it's going to stick together. On the other hand, if I then increase the extensile activity, keeping everything else the same, the extensile flows are essentially going to tear it apart because it's very active, it's very turbulent, the droplet does. Yes?
Actually try to do this simulation starting from homogeneous states rather than kind of phase separated.
Sorry?
Well mixed state instead of the phase separated one.
Sorry, we started with the fully mixed state, right? Those were the initial simulations, and we did the.
Actually going to the phase separated state at the end. If you start from fully mixed one.
If you will be this would be this setup, right?
Oh, yeah.
Because this, if it starts fully phase separated, if it starts fully mixed, it becomes essentially fully phase separated over time. And this is just sort of trying to understand why increasing extensile activity is tearing it apart, and trying to understand why increasing contractile activity is actually helping in spite of contractile activity not having any effect on the motion. And so essentially the idea is, if you start with both activities being fairly low, a droplet is going to stick together. If you then increase the extensile activity, the activity is going to tear apart the droplet because it becomes much more turbulent, much more active. But then if you increase contractile activity, the droplet again sticks together. If we're still at high extensile activity, but in some way, which we're not entirely clear about, contractile activity acts to adhere the cells together. So doing this in a slightly more measured way, here we have the highest component, the largest component of the droplet for two extensile and two contractile activities. And this plots the averaged out contractile, the largest component for contractile and extensile activities. And what you see is that at high contractile activities, it holds together much better than at low ones. And what seems to be the reason for this is, and this was already shown in the original paper, is that even if you have the passive vertex model in the fluid phase, the contractile activity renders it solid. So if you measure here its shear modulus, it's still going to be finite. And on the other hand, the extensile activity, even if you're in the solid phase of the vertex model, is going to make it fluid in terms of shear response. So essentially, what you're mixing is a fluid and a solid. Now, this doesn't seem to be an entirely passive effect, because if we start from different initial conditions, so that we have initially completely mixed state, and then three different fully phase separated states, which are just so that if it happens to be a curvature effect, we cut all three of them. And then you measure the sorting index in the 3 phase-separated states, it slightly decreases but not significantly. And in the mixed state, it goes up over time. But if you then look at the passive energy, it turns out that the energy of the phase-separated system is actually lower, of the mixed system is lower than the phase-separated system. And as it sorts, its energy goes up. So it's not just that there's an energy penalty associated with the barrier, because some cells are stretched and others aren't. Now, lastly, while we were doing this with the wet model, with the dry model, if you go to the wet model, if you do the whole internal dissipation scheme, the system still survives, you still get phase separation, and the optimal value of phase separation is roughly the same as in the dry model, but as you then increase extensile activity, here we're calling internal and external, the phase separation is less significant, which probably just comes from, well, if internal dissipation, you're going to get more pronounced flows over time. So that's it for my presentation. I want to thank everyone I worked with on this project, and thank you for your attention.
We have time for some questions. Now, can we go back to Ananya's question about the length scales of the active nematic? Can you tell us how to think about the active nematic in the context of the vertex model? I just compare and contrast to the way that we normally think about periodic array of vortices is that you're basically confining it on the active length scale, right?
Yeah.
And when I start allowing more than one vortex to be within the channel, I start getting a mess.
Yeah, it's broadly doing that because if you have length scale which are smaller than the width of the channel, or at least it doesn't map precisely the values, but that could just be because we also have like the cell size length scale going on. It's not entirely clear how to connect them. But as you reduce the length scale, it's going to go into first doing vortices. We haven't quite gotten it yet to make a very nice dancing state, but it starts doing channel-wide vortices, and then it goes into active turbulence. So essentially, it's doing the same thing.
Yeah. Same.
Friction is reducing your length scale, and then you go to the usual sequence of steps.
Thank you. Other questions? Yes, Silka?
So what you say, so even though you don't really have like the classic active coupling terms ultimately merging in here, so this is all, it's mostly the pair dissipation aligning them. Is this the origin of it?
Sorry?
But should I think of this as a disordered system which is getting flow aligned by the pair dissipation, or should I think of this as a broken symmetry phase?
I think it's essentially being flow aligned by, we're not completely sure, but it does seem to be just, if you had flows, it starts aligning, but if it were, if the activity was turned off, there's nothing keeping it aligned, essentially. Okay, thank you.
If not, let's thank the speaker one more time.